Hello everyone, my name is Sonali, presenting to you this video lecture as part of an initiative by DHE Haryana, and in continuation to the previous video lecture where I talked about morphological features of Pinus. In this following video I will talk about the anatomical features of the plant Pinus. The difference between morphology and anatomy is that the morphology tells us about the outer or superficial appearance or how the plant look from outside, whereas anatomy tells us what is inside the plant or organism in terms of what type of cells or tissue make that organism and its parts. Through this video, you will learn about what is the internal structure of Pinus, what type of cell or tissue makes its parts, what are their functions, and how they help plant grow. We will start with the anatomy of root with focus on primary root, secondary growth in it, and mycorrhizal root. Next, we will look into the anatomy of young pine stem, old pine stem, and at the end, anatomy of leaf of Pinus will be discussed, which is basically known as a needle. Young Pinus root is a simpler structure. When you cut it into a transverse section, you have three major regions: outer, middle, and central. Outer region is the dermal layer. It consists of epidermal cells which have root hairs. This layer is also known as piliferous layer for the pili-like appearance from root hairs. This layer is followed by a region of 4 to 5 layers of parenchymatous cells. These cells are filled with starch. On the inner side, this region is limited by single endodermis layer, which is brown colored as you can see in the diagram. Endodermal cells are water barrier cells that do not allow movement of water through the cell wall because of deposition of suberin. This deposition makes Casparian strip in endodermis. Water molecule has to pass through the cell to go to the central region. Endodermis with outer parenchymatous cells form cortex, a term generally used for middle region. Cells from epidermis to cortex outside endodermis are loosely arranged, and that is the reason water can travel through these spaces easily. But it is this layer of endodermis which is tightly packed that it does not allow molecules to enter the vascular tissue easily, generating concentration gradient, which in turn is important for the absorption of water and minerals from the soil. Now, in the center is the central region. It is also known as stele. It is made of several layered pericycle followed by resin ducts, vascular bundles, and a parenchymatous pith in the center. Vascular bundles are arranged along the radius separately, as you can see very well in the diagram. Each xylem and phloem tissue is on different radius, so they can be described as radial. Early formed xylem, which is smaller in size, known as protoxylem, is towards outside, and late formed xylem, which is large in size, known as metaxylem, is towards the center, as you can see in the diagram in dark blue color. This is known as exact condition. Xylem tissue here is Y-shaped and there can be 2 to 6 xylem tissues present in a vascular bundle depending upon the species. Here 3 xylem tissue are present, hence the condition is known as triarch. With each xylem tissue a resin duct is present. Vascular cambium and cork cambium, which are responsible for secondary growth in vascular bundles and cortex respectively, develop later. It is found that they develop from pericycle and some of cortex cells. One important point to note here is pericycle is also the layer which give rise to lateral roots while growing. Now, here is a microscopic picture of TS of old Pinus root to show secondary growth. Here you can see dark and compactly arranged cells. It is the outermost layer, periderm, which is formed by the cork cambium on the outer side. These cells are highly suberized and tanniniferous. Next to it are cortex cells inside. One thing to remember is that secondary growth takes place in the older regions of root where water absorption does not take place. Here, endodermal cells get crushed when new secondary cells are formed. In the central region, vascular cambium, which developed from pericycle, starts to form secondary phloem on outer side and secondary xylem on inner side in such a way that after some time the vascular bundles which were arranged radially make continuous rings of secondary xylem towards the center and secondary phloem on outer side. Primary phloem also gets crushed and pushed into the cortex region in this process, and the pith in the center gets lost with the time. Resin ducts still continue to exist. After talking about secondary growth in normal roots, let us take a look at mycorrhizal roots of Pinus. It is almost similar to young roots, only differences being secondary growth does not take place, resin ducts are absent, and it has fungi for symbiotic associations which grows into cortex region between intercellular spaces and form heart-egg net on outside. You can see in the diagram red thread-like structures represent the fungal mycelium and they extend to the cortex region. Endodermis is present, followed by a pericycle layer. In the central region, simple vascular bundles are present, arranged radially with a central pith. One thing to remember here is that vessels and fibers in xylem and companion cells in phloem are absent in Pinus, as are in most of the gymnosperms. So, when we will talk about xylem and phloem, here we will only talk about xylem tracheids and xylem parenchyma and phloem sieve cells and phloem parenchyma. Now, before ending the anatomy of roots, this is a closer view of microscopic section of mycorrhizal root. Here you can see large round cortical cells, and in intercellular spaces, thread-like structures which are fungal mycelium. Now let's move on to the next part of the lecture, that is anatomy of Pinus stem. We will start with the anatomy of young Pinus stem. On the left you can see a diagram of the transverse section of young stem of pine. I would like you to keep a look at it for better understanding. As in roots, the section of stem also is divided into three major regions: outer, middle, and central. Outermost layer is of epidermis. Walls of epidermal cells toward the outside are thick. Also, epidermal cells secrete cutin, which deposits just outside the cells and form thick cuticle. This cuticle helps in protecting the plant from external factors, dry conditions being one of them. Just beneath epidermis is hypodermis. As the name suggests, it lies beneath the epidermis. It is made up of sclerenchymatous cells, which are lignified dead cells and provide rigidity, and is usually 1 to 2 layered. Next is the middle region, made of cortex, endodermis, and resin ducts in between the region. Parenchymatous cells make major portion of cortex. In between these cells are present resin ducts. Ducts are lined with epithelial cells on inner side. Towards the center, single layered endodermis is present. And now coming to the central region, next to endodermis is pericycle. It is mostly single layered. Vascular bundles are next to it. They are conjoint, that is, xylem and phloem tissue are present together, not separately as was in roots. Collateral, that is placed lateral to each other along the radius, with xylem towards the center and phloem on the outer side. And open, that is, vascular cambium is present in between xylem and phloem. Xylem here is endark. Unlike roots, protoxylem is towards the center and metaxylem is towards the outer side. In young stem, xylem and phloem are present in bundles, each bundle separated by a row of medullary ray cells. As you can also see in the diagram, there are two bundles, and in between them along the radius is present a row of medullary ray cells. They are uniseriate, that is arranged in a single row. They are long, narrow cells and parenchymatous in nature. And at last, in the center is pith, made of parenchymatous cells, which gets filled with tannin over the time. Pith also contains resin ducts. Ray cells make a connection between pericycle and the pith. With this, we can say that in young pine stem, vascular cambium that is present between xylem and phloem is not continuous and is disturbed by ray cells. So here comes the question: How does secondary growth takes place where ray cells are present? We will talk about it under the next topic, which is secondary growth in pinus stem. Before that, a quick point to mention is that the xylem in pinus, or the wood formed by xylem in pinus, is pycnoxylic. That is, parenchymatous cells are fewer in comparison to trachees, which makes it a very compactly and neatly arranged xylem tissue. This is known as pycnoxylic condition and is characteristic feature of pinus. Now, here is a microscopic picture of transverse section of a young pinus stem. Here you can see the outline is irregular or wavy. This is because of scale leaves. Where scale leaves are present, the outline is depressed. Next, you can see parenchymatous cortex with resin ducts arranged in a circle. As you move inward, dark bluish colored phloem cells are visible, and near to them lie brown colored xylem. In the center, pith is present, and in this whole section you see red colored cells or spots in the cell. In cortex and pith, they are tannin filled cells. Now, this is a microscopic picture of pine stem where secondary growth has started. Older tissues are getting crushed to make space for the new ones. Next to cortex is almost a continuous ring of secondary phloem, and on the inner side, secondary xylem is being added. Here you can see single rows of medullary ray cells along the radius extending from outer side to the pith. They are easily visible as blue colored lines in between secondary xylem. Now, this is a diagram showing transverse section on old stem of pinus. Moving from outside to the center, you can see cork on the outside, followed by cork cambium and secondary cortex cells. Cork cambium is not present from the start but formed later by cortex cells and produce cork towards outer side and secondary cortex cells towards inner side. This leads to the crushing of epidermis and hypodermis. The other cambium is the vascular cambium. It is present in vascular bundles from the start and is of primary origin, formed by procambium, unlike cork cambium, which is of secondary origin. Now here we will talk about the point, how does cambium leads to secondary growth in all of the vascular cylinder when it is present only in between xylem and phloem and not in medullary rays. The answer lies in the fact that there are two types of cambium: fascicular cambium, also known as fusiform initials, They are present between xylem and phloem tissue and form secondary xylem and secondary phloem. And second one is interfascicular cambium, also known as ray initials. They are present in medullary ray cells and form secondary ray cells. When secondary growth takes place, both types of cambium join and form a continuous ring and produce secondary xylem and secondary phloem, where xylem and phloem are present, and medullary rays where ray cells are present. Now, another interesting fact about pine stem is the different rate of activity of cambium in different seasons, depending upon the growth affecting factors like temperature, water, sunlight, etc. So, what happens is that when the conditions are difficult, like in winters, the growth activity of plant slows down. As a result, cambium activity also slows down. This leads to the formation of xylem cells, which are smaller in size, and when the conditions return to normal, like in spring, the growth rate of plant increases and cambium divides actively, forming large xylem tracheids for better water transport, which in turn further enhance the growth of the plant. Now, this different rate is seen as pattern in the transverse section of old stem as light and dark rings where xylem is formed, that is the wood region. And this one light and one dark ring of wood forms one annual ring representing spring and winter season of an year. Every year these rings are formed and can be seen clearly in the diagram also. These rings are often used to calculate the age of the plant. One light and one dark ring is equal to 1 year of age. Here again is a microscopic picture showing transverse section through an old Pinus stem. You can see the different cells labeled. Also, you can see two annual rings, which very much suggests that the plant is 2 years old. And in this picture, more annual rings are present, and I would like you to guess the age of the plant. Now, here is a picture of three types of sections through old Pinus stem. In left is a simple transverse section of wood, that is secondary xylem, which you can easily identify. Next to it is RLS, that is radial longitudinal section, and to the right is TLS, that is tangential longitudinal section. These sections show xylem tracheids, resin ducts and medullary rays from different angles. Now, moving on to the last topic of this lecture, anatomy of the leaf, which in case of Pinus is commonly known as needle because of its needle-like shape. When we cut the needle transversely, we see a triangular structure with three major regions: outer, middle and central. Outermost layer is of thick walled epidermis, which also secretes cutin on the outer side, and this forms a protective layer on the surface as thick cuticle. Sunken stomata are present. The layer just beneath the epidermis is hypodermis. It is 2 to 3 layered sclerenchyma cells. All these features are xerophytic, which prevent water loss and protect the plant or leaf from harsh conditions like winters and high temperature and sunlight, etc. Next is the middle region. Large chlorenchymatous cells form major part of the region, also known as mesophyll. It is homogeneous in nature, that is, only one type of cells are present in mesophyll, unlike angiosperms where mostly two type of mesophyll cells are present: spongy parenchyma and palisade parenchyma. Now, one peculiar feature of chlorenchymatous mesophyll cells is that the inner wall of the cell folds inside the cell and form peg-like infoldings. Resin ducts are present in this region. Position of resin ducts may vary from species to species, that is, they can be present towards the hypodermis or endodermis or in between mesophyll cells. Endodermis is the inner layer of middle region, and point to note here is that endodermal cells lack Casparian strips in needles. Next is the central region, which has layer of pericycle on the outer side. It is multi-layered parenchymatous and contains transfusion tissue and tracheids, which help in better movement of water from the vascular bundles to the mesophyll cells. Vascular bundles are endark, that is, protoxylem is present towards the center and metaxylem is towards the outer side. Vascular bundles are conjoint, collateral and open like in stem. Yes, the condition is open, that is, cambium is present, but secondary growth is not that prominent. Now, number of vascular bundles in a needle vary from species to species. Basically, two types are present: haploxylon and diploxylon. In haploxylon, single vascular bundle is present and is surrounded by sclerenchyma cells, whereas in diploxylon, two vascular bundles are present, and in between them, sclerenchyma cells are present as a T-shaped patch. And since there are two vascular bundles present in this diagram, it's a diploxylon condition. Phloem tissue will be present on the outer side and xylem is present on the inner side. The inner side here means the side which is facing towards the central axis of the spur, where more than one needles are present. In this picture, you can understand what that central axis of spur is. First is on monofoliate and is haploxylon. Others have a central axis around which all the needles are arranged. Bifoliate spur have an axis in between them. In the center of trifoliate spur, there is a central axis, and similarly, pentafoliate also has a central axis. So, in all spurs except for monofoliate, vascular bundles have xylem towards the center of the spur and phloem towards the outer side. Also, one point to remember here is both monofoliate and pentafoliate are haploxylon, whereas bifoliate and trifoliate spurs are diploxylon. Pentafoliate spur has much narrow spurs compared to the diameter of spur with 2 and 3 needles, hence haploxylon condition suits it better. Now, here is a closer look at outer and middle region. You can very well see outermost thick cuticle, epidermis. Hypodermis is a bit difficult to see, but resin ducts are visible. Mesophyll cells are also visible, and their peg-like infoldings are also visible. Point to note is that these infoldings increase the inner wall surface area and hence increases the area for arrangement of photosynthetic enzymes and complexes, hence compensate the reduction in surface area because of narrow needle shape of the leaf of pine. And this is a microscopic picture of haploxylon condition in needle with its different parts labeled. I would like you to have a look at it. So now, coming to an end of this lecture, I would like to share a small exercise for you all. Let's see if you can identify the given section and how many of its parts or cells can you name? Interesting, right? Hope you now understand the structure of Pinus plant much better. Thank you for watching.